Okay, so it's 6:10. So we'll be starting today's session. Hello, am I audible? Can someone let me know if I'm audible? Hello. 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 Hello, am I audible? Yeah, I guess I'm audible. Yeah, so it's 6:10. So I had some network issues on my system. So I'll share my screen now. Give me a moment. Yeah. Yeah. So good evening, everyone. Good evening, everyone. Sorry. Good evening, everyone. My name is Aishi and I'm a PMRF research scholar from the Department of Chemistry, IIT Bombay. So, welcome to the 11th week live session. Basically, live session of this course, post pathogen interaction immunology, taken by course instructor Professor Himanshu Kumar from IISER Bhopal. So basically what we do in these live sessions is that we pick some old year, previous year assignment questions, old assignment questions, and we try to solve it. And we not only discuss the answers to those questions, but we also try to discuss the concepts behind those questions so that it's easier for us to solve questions following the same concept. So that it basically prepares us for competitive exams as well, right? So that is how we go about it. Now we try to keep these sessions as interactive as possible. For example, I speak aloud the question, then I'll give you a few minutes to tell me the answer if you know, but you have to give me proper explanation, not just the answer. Why that answer and why not the other options, and then we'll move on to discussing the concepts behind that, discussing basically the concept behind that answer, so that you do not only know the answer to that question, but you also know that you will also be able to solve similar type of other questions, right? That is what we will be doing. Yeah, that is what we will be doing. Give me a moment, there is some network issue. One second, I'm just trying to figure out the network issue. Yeah. So, and if you can we know the answer, then you just write in the chat box or maybe unmute and let me know. At the end of each question also, I'll give you a few minutes to ask me queries or doubts if you have regarding that question and why that particular answer is the correct answer. If I know the answer at the moment, I will surely answer you then and there. If I don't, then sorry, you have to give me some time. I'll read about it and let you know definitely by the next class. Okay, so that is basically how we conduct these live sessions. Okay, so with this background, we'll move on to the first question for today. Yeah, so the first question is on your screen now. Sorry. Yeah, so yeah. So the question is, what are some of the diverse applications of interferons in medical treatment? You can choose multiple, like it's an MSQ, multiple select question. Multiple choices are correct. So the options are A, treatment of viral infections, B, cancer therapy, C, increasing insulin sensitivity, D, immunotherapy, E, stimulating stem cell differentiation, F, healing bone fractures, G, neurological conditions like multiple sclerosis, and H, reducing neuroinflammation. Which one do you think is the correct answer? Wait for a few minutes. Anybody who would like to try and answer the question? There's no harm in trying. Wait for a few minutes and I'll move on to the Anybody? Okay. So okay. So we'll discuss the answer now. So these are the correct answers, right? So interferons are basically cytokines with well-established roles in antiviral defense, immune modulation, and certain therapeutic areas. Now let's go through the options carefully. Treatment of viral infections. It's correct because interferons, especially interferon alpha, they are used for viral infections like hepatitis B and C. Now, second option is cancer therapy. That is also correct because they have anti-proliferative effects and have been used in cancers like melanoma and some leukemias. Now, option C, increasing insulin sensitivity, is incorrect because interferons are not used for this. In fact, they can sometimes worsen glucose metabolism. Then option D, immunotherapy, that is correct because they modulate immune responses and are part of immunotherapeutic strategies. Then option E, stimulating stem cell differentiation, incorrect. It's not a standard or clinically established role. Option F, healing bone fractures, also incorrect because no established therapeutic use here. Option G is neurological conditions like multiple sclerosis. Correct? Because interferon beta is the standard treatment for multiple sclerosis. Then we have option H, which is reducing neuroinflammation, which is also correct because particularly it is relevant in multiple sclerosis where interferon beta reduces inflammatory processes in the central nervous system. Right? So, what are interferons? How were it discovered? Let's go back into history once. So, it was discovered in 1957 by Isaacs and Lindenman. They did an experiment using chicken cell cultures, like found a substance that interfered with viral replication and hence were termed interferon because they were interfering with something. So, very easy, they just coined this name. Okay. So, now if we go into the detail, what are interferons, what is their role? Basically, they are nothing but naturally occurring proteins and glycoproteins. Okay, they are secreted by eukaryotic cells. They're secreted by eukaryotic cells and in response to viral infections, tumors and other biological inducers. They produce clinical benefits for disease states such as hepatitis. Then we have this various cancers. Then we have multiple sclerosis that I just discussed, and many other diseases. Structurally, they are part of the helical cytokine family, which are characterized by an amino acid chain that is 145 to 166 amino acids long. So what is the general action of interferon? So interferons are small proteins released by macrophages, lymphocytes, and tissue cells infected with the virus. So when a tissue cell is infected by a virus, it releases interferon. Interferons will diffuse to the surrounding cells. When it binds to receptors on the surface of those adjacent cells, they begin the production of a protein that prevents the synthesis of viral proteins. So this prevents the spread of the virus throughout the body. Okay, so you can see over here, host cell is infected by virus. Okay, if you can see over here, this virus infection is there. Interferon virus is making new viruses, and new viruses are going and affecting more cells. Now, because of the viral infection, the interferon genes are turned on. The DNA, mRNA is produced, and interferon molecules are produced. Okay. Now, these interferon molecules, they enter into host cell 2 from cell 1, and they induce changes that protect the cells. They make some kind of changes to the system so that antiviral proteins are produced that protect the cell from viral reproduction. So type 1 interferons can be alpha and beta. Alpha interferons are produced by leukocytes. Beta interferons are produced by fibroblasts. Now both bind to interferon cell receptors type 1, and both are encoded on chromosome 9. Now they have different binding affinities but similar biological effects. So viral infection is a stimulus for alpha and beta expression, and it is used to basically mobilize our first line of defense against invading organisms. So largest group, and it's the largest group of interferons and are secreted by almost all cell types. Okay. Then we have differences between these two types of interferons. Interferon alpha and interferon alpha and beta, and also interferon gamma. Interferon alpha and beta type 1 interferons, interferon beta gamma is type 2. Interferon alpha and beta are acid stable. Type 2 interferons are acid labile. Type 1 are like 14 interferon alpha genes and 1 interferon beta gene, whereas type 2 means 1 interferon gamma gene. Type 1, they basically act by binding a common interferon alpha beta receptor. Type 2 acts by binding an interferon gamma receptor. Type 1 encoded by chromosome 9, type 2 chromosome 12. Type 1 interferon is produced by almost all types of cells, alpha majorly by leukocytes and beta by fibroblasts. Type 2 is immune cells like T cells, natural killer cells, B cells, and APCs. Okay. Then type 1 can induce antiviral state. They can upregulate MHC1. MHC1 is the major histocompatibility complex that we have discussed in previous sessions. And type 2 basically it's an immunomodulator, upregulates MHC1 and 2 expression and CMI response. Okay. Then we see type 1 is a stimuli, lethal stimuli, like it's a viral infections. Type 2 is immune and inflammatory stimuli. One second. Immune and interferon stimuli. Now, if we talk about the type 2 interferon, which is basically gamma, it basically binds to type 2 receptors and its genes are encoded on chromosome 12. They initially believed that T helper cell type lymphocyte, cytotoxic lymphocytes and natural killer cells only produce interferon gamma. Okay. Now evidence tells that B cells, natural killer T cells and professional antigen presenting cells also secrete interferon gamma. Okay. Gamma production follows activation with immune and inflammatory stimuli rather than viral infection. As we discussed for interferon 1, which was a stimuli like viral infection. And its production is controlled by cytokines interleukin, -12 and 18. Now, interferon gamma is also called the immune interferon. It is activated by activated T cells or natural killer cells, and interferon gamma promotes inflammation and regulates MHC class 1 and class 2 molecules. It is unstable at pH 2 and it is advantageous during host cell infections when the virus switches off the RNA and protein synthesis so that IFN alpha and IFN beta are prevented from being made. So IFN gamma appears either late in the infection or stimulation of the immune system. Now, what is the mode of action of the interferon? The mode of action of interferon can be divided into 2 stages. Number 1, induction, which results in release of interferon, and number 2, antiviral state production in other cells where they release interferon. Okay. Induction stage is induction of interferon in human cells is controlled by chromosome 9, right? So all multiplying viruses induce interferon and it's believed that double-stranded RNA is the specific inducer. Free interferon initiates an antiviral state in other cells by binding to a receptor on their cell surface. Now, if we talk about the antiviral state, the second stage represents the creation of an antiviral state in the neighboring cells by the released interferon binding to the interferon specific receptor on the cell surface. Now, in man, chromosome 21 is required and perhaps calls for the synthesis of interferon receptor molecules. The antiviral state has 2 modes of action: dsRNA, double-stranded RNA, in the presence of interferon-induced antiviral state. It stimulates the phosphorylation of certain cellular proteins, resulting in number 1, impairing their function in the initiation of protein synthesis. Number 2, double-stranded RNA activates ribonuclease, which degrades mRNA and hence also stops protein synthesis. Now, the mode of action, if you see virus infection, double-stranded RNA is the synthesis of interferon, and there is a transcriptional activation of interferon genes, which leads to synthesis of interferon and secretion of interferons. It binds to the interferon receptor on the next cell or the adjacent cell, and induces the transcriptional activation of antiviral genes, and which leads to the synthesis of antiviral proteins, which leads to blocking of the viral infection in the second cell. Okay, not good. Now, if we talk about interferon in terms of therapy as a drug, then interferon was scarce and expensive until 1980, when the interferon gene was inserted into the bacteria using recombinant DNA technology, allowing mass cultivation and purification from bacterial cultures. It can also be commercially produced using yeast. Now, it's used in combination with chemotherapy and radiation as a treatment for cancers and treatment and control of multiple sclerosis. Pegylated interferon alpha in combination with the antiviral drug ribavirin is used in the treatment of hepatitis C. Prevention and treatment of viral respiratory diseases such as cold and flu, and effective against tumors like melanoma and Kaposi sarcoma. Now, what are the side effects of interferons? Common side effects include fever, fatigue, muscular pains, local inflammation, convulsions, dizziness, hair thinning, erythema, and high levels of interferons can cause kidney, liver, bone marrow, and heart toxicity. Now, interferon therapy is only effective if started before the virus is inoculated and is thus not applicable to natural viral infections. Now, what is the route of administration? So, IFN alpha and IFN gamma are mostly administered by intramuscular injection. Now, the injection of interferons in the muscle and in veins or under the skin is generally well tolerated. Now, what are the interferon drugs? Interferons are broken down into recombinant versions of specific interferon subtype and purified blends of natural human interferon. That's the mechanism of the interferon therapy that has been going on. So many of these are in clinical use and are given intramuscularly or subcutaneously. Okay. Recombinant forms of alpha interferon include alpha 2A, drug name Roferon, alpha 2B, drug name Intron A, alpha N1, drug name Velferon, alpha N3, drug name Aferon, and alpha Con1, drug name Infergen. So, recombinant forms of beta interferon include basically beta 1A drug named Avonex, beta 1B drug named Betasteron. Recombinant forms of gamma interferon include gamma 1B drug named Asimune. Now, receptor for interferons in vaccinia virus inhibits the interferon's ability to function. So, H5N1 influenza virus known to cause bird flu has resistance to interferons and other cytokines due to a single amino acid mutation in its non-structural protein NS1, and this is the reason for its high mortality rates. Now, concluding, interferons have overlapping but different biological activities. Their mechanisms of action are not yet fully understood. Therefore, there is a lot of room for future growth within this field. Interferon-based strategies can possibly further tailor to each individual patient according to early response dynamics. And other immunomodulators that are being tested include Zaraxin and Ceplin. Now, if we talk about viral resistance to interferons, some viruses have manifested methods that give them a way around interferons alpha-beta response. So, the methods are inhibition of interferon signaling and production, or blocking of the function of the interferon-induced proteins, or Japanese encephalitis virus blocks interferon alpha signaling, or pox viruses encode basically a soluble interferon receptor homolog that acts as a decoy to inhibit the biological activity of interferons, like BI8R protein acts as a. So now, if you talk about the pharmacology of interferon, there are two types: either natural interferons produced in the body or man-made interferons that are recombinant. So, as we said, let's come back to the basics. It plays an important role in the first line of defense against viral infections. They are a part of the non-specific immune system, and interferons are made by cells in response to an appropriate stimulus. Now, alpha is leukocyte interferon produced by virus-infected leukocytes. Beta is fibroblast interferon produced by virus-infected fibroblasts or epithelial cells. And gamma is immune interferon produced by certain activated T cells and natural killer cells. So, we have already discussed how the virus infection happens and how interferon production from one cell saves the other cell from viral infection, right? So IFN alpha and beta will be their mechanism of action is induction of inhibitory protein synthesis. IFN gamma is including class 2 MHC molecules of APC antigen presenting cells and including increasing ability of macrophages to resist viral influx and kill other cells if infected. All interferons in general increases class 1 MHC molecules and activity of natural killer cells. Now the viral defense against interferons. So they block interferon binding to cells. They inhibit action of interferon-induced protein kinase. They inhibit natural killer function. They interfere with cell surface expression major histocompatibility complex. And they can block complement activation and prevent apoptosis in the host cell. Now, what are indications for interferon? Okay. So, alpha is hepatitis B and C, if we talk about that. Hairy cell leukemia. Give me a moment. Okay. Then chronic myeloid leukemia, multiple myeloma, low-grade lymphomas, Kaposi sarcoma, melanoma, and beta will be multiple sclerosis, which is ulcerative colitis. Okay. So, gamma is chronic granulomatous disease, chronic myeloid leukemia, renal cell carcinoma. Now, this is the incomplete list of interferons that we're talking about. Now, if we talk about hepatitis virus epidemiology, it's a leading cause of HCV epidemiology. Leading cause of liver disease, HCV infection leads to decompensated. If we talk about what do we mean by HCV infection and HCV epidemiology, so it is a leading cause of hepatitis C is a viral infection caused by hepatitis C virus that primarily targets the liver. It's an RNA virus that infects hepatocytes, liver cells. It can be acute, like short-term, often asymptomatic, or chronic, like long-term, develops in 70 to 85% of the cases. And clinical significance is that chronic infection can lead to liver fibrosis, cirrhosis, hepatocellular carcinoma, like liver cancer. The transmission is mainly through blood-to-blood contact, such as contaminated needles, unsafe medical practices, blood transfusions, especially before screening was routine, and less commonly through sexual contact or from mother to child. And if we talk about the HCV epidemiology refers to the distribution, determinants, and patterns of the infection in populations. The key aspects of HCV epidemiology is that global burden, 50 to 60 million people worldwide are chronically affected. Higher prevalence in regions like parts of Africa, the Middle East, and South Asia. High risk groups are people who inject drugs, healthcare workers, needle stick injuries, individuals with unsafe transfusions or medical procedures. Now, there is a decline in transfusion-related cases due to screening and risk rise in cases linked to injection drug use in some regions. There are multiple viral genotypes with different geographic distributions and treatment responses. Okay, so interferon in hepatitis C, basically you can talk about the monotherapy, which is a standard interferons 3 million units combination therapy, standard interferon with ribavirin, and pegylated interferon with ribavirin. The limitations are monotherapy is not very effective, it's a cumbersome dosing and multiple side effects. Now, flu-like symptoms, headache, nausea, vomiting, diarrhea, depression, irritability, anxiety, injection site, reactions, partial alopecia, hematologic abnormalities, autoimmune disorders, all of these are interferon side effects from hepatitis C data. Now, if we talk about pegylated interferon, so if we talk about pegylated interferons, basically it's an ideal setting for PEG interferon. Polyethylene glycol and interferon ratio has to be maintained. Now, pegylation was developed to overcome disadvantages of standard interferon. It shields interferon from enzymatic degradation, thus lower systemic clearance. It allows less frequent dosing and it achieves higher sustained serum interferon. Okay, speg interferon. Interferons are important in the non-specific immune response. They're effective in the treatment of patients with chronic hepatitis. Pegylated interferons are superior therapies for patients with HCV, and side effects should be monitored closely. Now, what is the immune response to virus infections? As I said, viruses often modify the cells within which they replicate, thereby rendering them foreign and susceptible to attack by sensitized lymphocytes. Furthermore, some viruses can multiply within lymphocytes and mononuclear phagocytes that are important components of immunological defenses. A good example is HIV, which attaches to and destroys the CD4 plus helper lymphocytes that are so important to the integrity of the immune system. However, the animal world has developed efficient and highly complex mechanisms for combating infection by parasites, which can be considered under the headings of innate immunity and adaptive immunity. By innate, we mean those defense mechanisms with which we are born and form the first line of protection against microbial invasion. They can be regarded as built-in defenses and fall into two categories: those that protect the individual and others genetically mediated that determine the resistance to susceptibility of populations. Okay, we have talked about all of these. Now we need to get into detail. Now, if you talk about apoptosis, it's also known as programmed cell death, which is the ubiquitous mode of cell death known to be responsible for clearance of unwanted, injured, or virus-infected cells. Now, cells undergoing apoptosis are accompanied by characteristic morphological changes, including cell shrinkage and deformation, chromatin condensation, nuclear fragmentation, and plasma membrane blebbing, which forms the apoptotic body containing the fragments of the cytoplasm or the nucleus. Now, in contrast to necrosis, apoptotic cells form apoptotic bodies. Now, what are apoptotic bodies? Basically, they are phagocytosed by the neighboring cells without the release of cellular contents. Now, apoptosis plays important roles in physiology and pathology and can be triggered by numerous stimuli, including ischemia, hypoxia, exposure to certain drugs and chemicals, immune reactions, infectious agents, high temperature, radiation, and various disease states. Now, if we talk about the local non-specific defenses, soluble factors such as the interferons, complement, and C-reactive protein, and phagocytic cells, particularly natural killer cells, all of which are important components of the innate immune system. The various elements of these responses can be mobilized very quickly within a matter of hours. Now, when the pathogen enters the skin, confronted with macrophages and other phagocytic cells possessing microbial sensors like TLRs, like the toll-like receptors, a family of evolutionary conserved pattern recognition systems that, among other activities, enhance the expression of MHC molecules and are thus important in the innate immune response. Now, these interactions between cells of the immune system are mediated by substantial variety of small glycoproteins referred to collectively as cytokines. Now, this is an umbrella term that encompasses a wide range of molecules. Cytokines act as chemical messengers, stimulating or inhibiting the activities of the various cellular components of the immune system. As such, they form an important component of innate immunity and can provide links between this and certain of the adaptive components. They contain several subgroups, include interferons. Other categories include the chemokines, which act as chemical attractants of leukocytes to sites of inflammation. Now, there are three classes of cytokines: pro-inflammatory, anti-inflammatory, chemokines. Initially function locally in antiviral defense, in larger quantities enter circulation, have global effects. A localized viral infection produces global effects. Now, inflammation usually stimulates potent immune responses. Cytopathic viruses can cause inflammation because they promote cell and tissue damage and activate the innate response. Consequently, cytopathic viral genomes encode proteins that modulate this immune response, like adenoviruses, herpes viruses, pox viruses. Now, some viruses do not stimulate inflammation. Typically, non-cytopathic viruses, like cells are not damaged, no apoptosis and necrosis, lower ineffective innate immune response, do not effectively activate adaptive immune response. Now, non-cytopathic viruses have dramatically different interactions with the host immune system. They have persistent infections, rarely are inefficiently cleared. As we said, the interferons are a member of a large group of proteins called cytokines, which affect a wide range of target cells and tissues by binding to specific receptors present on the surface of target cells. Right? Interferons play an important role in the first line of defense against viral infections. They are part of the non-specific immune system and are induced at an early stage of viral infection, before the specific immune system has had time to respond. Now, interferons are not expressed on normal cells. Are not expressed on normal cells, but virus infection of a cell causes interferons to produce and release from a cell. And the cell will often eventually die as a result of infection. Now, the interferons then bind to the target cell and initiate an antiviral state. Now, there are several interferons, as I said, which differ in the way they are produced in chemical composition and in mode of action. They are proteins with molecular weights of about 20 kilodalton manufactured by leukocytes or fibroblasts in response not only to viral infection but also to stimulation by natural or synthetic double-stranded RNA and some bacteria like chlamydia. Now, these molecules are not virus specific, so the interferon induced by one virus is effective against others. On the other hand, they are species specific, so the interferon produced by a guinea pig is ineffective in mouse or human cells. Interferon G differs in a major way from alpha and beta. Like gamma differs in a major way from alpha and beta. Varieties as it is produced by a subset of T memory cells in response to stimulation by an antigen previously encountered. So interferons do not really kill viruses, nor do they act like antibodies. The production of interferon alpha beta is rapid. Within hours of infection declines by 10 hours. Interferon binding to interferon receptors leads to synthesis of greater than 1000 cell proteins. And mechanisms of most of these are not known. Now, what is the mode of action of these interferons? Both DNA and RNA viruses can induce interferon. Production of viruses are target specific, and then DNA or RNA virus binds to specific receptors on a target cell. It induces transcription of 20 to 30 genes, ultimately forming mRNA, and this induces formation of 20 to 30 proteins or interferons. Among these proteins, three proteins appears to play important role in an induction of antiviral state. First protein is called 2,5-oligoasynthase, results in activation of second protein called ribonuclease, which can break down mRNA and causes expression of third protein, a protein kinase. The kinase inhibits the initiation step of the protein synthesis. Now, these activities of proteins target not only viral protein synthesis but also of the host protein synthesis. Activation of these proteins results in death of cell, but at least the progression of virus infection is prevented. Now, the interferon system is dangerous. Interferon induces the expression of many deleterious gene products. Most of our cells have interferon receptors. Now, interferons have dramatic physiological consequences: fever, chills, nausea, malaise, and every viral infection results in interferon production. One reason why the flu-like symptoms are so common. We have talked about the complement in one of the previous classes, but just to summarize, the complement system functions as an immune surveillance system that rapidly responds to infection. The activation of the complement system typically occurs via three distinct target recognition pathways: the classical, lectin, alternative. And the complement system has been shown to exhibit numerous antiviral mechanisms via direct utilization of both enveloped and unenveloped viruses and on the promotion of other immune responses. Not going into too much detail. Okay. So now we talk about cytokines, which are basically nothing but the key communicators of the immune system. So they're proteins that help immune cells communicate and coordinate their responses. So they basically they guide immune cells to the site of infection or injury. They activate immune cells, increasing the ability to fight pathogens, regulate inflammation, ensuring the immune system response is balanced. And if you talk about chemokines, specialized cytokines that guide immune cells to specific sites. Example is CXCL8. It attracts neutrophils to infection sites, basically. Cytokines can be described by their functions. I have already discussed this in a previous class. I am not going into too much detail. These are their key features: receptor specificity, high affinity, signal transduction. So, a single cytokine can have different effects on different target cells. That is why it is pleiotropic. Like IL-4 promotes B cell activation but also inhibits macrophage activation. Cytokines can adapt their effects based on cell type and its environment. Then redundancy, different cytokines can have similar functions like IL-2, IL-4, IL-5 all promote B cell proliferation. Redundancy ensures that the immune system can function even if one cytokine is absent. Synergy, two or more cytokines work together to enhance their effects, like IL-4 and IL-5 together promote IgE production more effectively than either alone. Cytokine teamwork amplifies immune responses. Then we have antagonism. One cytokine inhibits the effect of another. Interferon gamma inhibits IL-4 to block B cells switching to IgE. The key concept is antagonism helps balance immune responses to prevent overactivation. Then we have cascade induction. One cytokine induces the production of additional cytokines. The IL-1 stimulates IL-6 production. Key concept is cascade induction amplifies and extends the immune responses. We will not go into too much details because we have already discussed in previous sessions about cytokines. So coming back to the question that we started with, what are some of the diverse applications of interferons in medical treatment? The options are as follows: treatment of viral infections, cancer therapy, immunotherapy, neurological conditions like multiple sclerosis, and reducing neuroinflammation. Anybody has any question? Anybody? If you have any question, you can ask me. I'll wait for a few minutes for you to ask questions and we'll move on to the next question for today. Anybody, any questions? Okay, so if you're done and you don't have any questions, we'll move on to the next question for today. How has genetic engineering impacted cytokine therapy? Option A. It has decreased the production of cytokines for clinical use. Option B. It has slowed the development of antiviral therapies. Option C. It has made it feasible to produce essential proteins for therapeutic purposes. Option D. It has eliminated the need for interferons in treating viral infections. And option E. It has only affected cytokines unrelated to interferons. You have to answer with proper detail explanation. आज मैं जब रील्स स्क्रॉल कर रही थी ना Anybody? यहाँ पर हमारा Anybody would like to answer? Anybody would like to answer? I'll wait for 2 more minutes and I'll move on to the discussion part. Okay. So since I'll go and I'll discuss the answer. So the right answer for this question would be yeah. So it would definitely be option C. It has made it feasible to produce essential proteins for therapeutic purposes. So genetic engineering or the study of recombinant DNA technology has had a transformative impact on cytokine therapy, especially for molecules like interferons, interleukins, and growth factors. Now, why option C is correct? Before genetic engineering, cytokines were difficult to isolate in large quantities from natural sources. With recombinant technology, genes encoding cytokines like interferons can be inserted into host systems like E. coli or mammalian cells. This allows large-scale, pure, cost-effective production of biologically active cytokines. As a result, many cytokines are now widely used clinically for antiviral therapy, cancer treatment, and immune modulation. For example, recombinant interferons are used in treating infections like hepatitis C and diseases like multiple sclerosis. While the other options are incorrect, option A, decreased production of cytokines, is incorrect because genetic engineering has increased, not decreased cytokine production. Option B, slowed antiviral therapy development, incorrect. It has actually accelerated the development of antiviral and immunotherapies. Option D, eliminated the need for interference, incorrect because interference is still really important. Although newer therapies may complement or replace them in some cases. Option E is also incorrect, only affected cytokines unrelated to interference, incorrect because interferons are among the most important cytokines produced using genetic engineering. Now, what is genetic engineering? If we start with the basic concept of that, we'll start with the first question, first doubt that arises in this question is what is genetic engineering, right? So, let's discuss that first. Yeah. So, if we talk about genetic engineering, basically it is also known as recombinant DNA technology, which means altering the genes in a living organism to produce a genetically modified organism, like GMO, with a new genotype. Now, various kinds of genetic modification are possible, inserting a foreign gene from one species into another, forming a transgenic organism, altering an existing gene so that its product is changed, or changing gene expression so that it is translated more often or not at all. Now, what are the basic steps of genetic engineering? Isolate the gene, insert it in a host using a vector, produce as many copies of the host as possible, and separate and purify the product of the gene. So basically, this is how you can isolate the gene using restriction enzymes or reverse transcriptase, basically mRNA converted into cDNA, which is a complementary strand produced using DNA polymerase. Advantage is more RNA in cell than DNA. Then inserting gene into vector, molecule of DNA which is used to carry a foreign gene into a host cell. It's a cloning vector. Inserting vector into host, and then we can do replica plating basically to talk about those particular colonies which will have the selectable marker, hence the converted or the transformed colonies. Now, multiplication of the host cells by cloning, like large-scale fermenters by cloning or genetically identical because of asexual reproduction. Extraction of the desired gene product. Now, DNA is a very large molecule made up of smaller units called nucleotides, and each nucleotide has three parts: a sugar, ribose, a phosphate molecule, and a nitrogenous base. Now, DNA polymerization only occurs in the 5' to 3' direction, and the nitrogenous base is the part of the nucleotide that carries genetic information. And the bases found in DNA are 4: adenine, cytosine, guanine, and thymine. Now, base pairing in RNA in place of thymine, we see uracil. Now, base pairing is accomplished by hydrogen bonding between the DNA base pair. The Watson-Crick base pairing indicates the base pairs in DNA are AT, 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between GC, guanine and cytosine. Now, a gene is a stretch of DNA that codes for a type of protein that has a function in the organism. It is a unit of heredity in a living organism. All living things depend on genes. Now, genes hold the information to build and maintain an organism's cells and pass genetic traits to offspring. Genes contain exons, which is a set of coding regions, and introns, which are non-coding rubbish regions, removed sequence, and are therefore labeled split genes. It is in a technique called splicing. Now, genome, the genetic complement of an organism, including all of its genes, is represented in its DNA. Now, gene expression is the process by which information from a gene is used in synthesis of a functional gene product. It is a protein. The process of gene expression is used by all known life, eukaryotes, prokaryotes, and viruses, to generate the macromolecular machinery for life. Now, if we talk about steps of gene expression, first there is transcription, mRNA synthesis, then post-transcriptional process, RNA splicing. Then we have translation, protein synthesis, then post-translational modification of a protein. Now, mutations are changes in the DNA sequence of a cell's genome caused by radiation, viruses, transposons, and mutagenic chemicals. The exchange of corresponding DNA segments between adjacent chromosomes during the special type of cell division that results in production of a new genetic makeup is called recombination. Now, in genetic engineering, recombination can also refer to artificial and deliberate recombination of pieces of DNA from different organisms, creating what we call is recombinant DNA. Genetic engineering is also known as a genetic modification, and is a process of changing the DNA or the genetic component in one's organism's genome. Now, coming to one of the most important part of genetic engineering, which is DNA cloning. Now, DNA cloning is a molecular biology technique that makes many identical copies of a piece of DNA, such as gene. In a typical cloning experiment, the target gene is inserted into a circular piece of DNA called a plasmid. The insertion is done using enzymes that can cut and paste DNA, and it produces a molecule of recombinant DNA or DNA assembled out of fragments from multiple sources. And the plasmid is introduced into bacteria via a process called transformation, and bacteria carrying the plasmid are selected using antibiotics. Now, bacteria with the correct plasmid are used to make more plasmid DNA, or in some cases reduced to express the gene and make protein. Restriction enzymes are DNA cutting enzymes. Each enzyme recognizes one or few target sequences and cuts DNA near those sequences. Now, many restriction enzymes make sticky or cohesive ends, producing ends with single-stranded DNA overhangs. However, some produce blunt ends. So, if two DNA molecules have matching ends, they can be joined by the enzyme DNA ligase. And DNA ligase is nothing, it's basically a glue, molecular glue. It's a DNA joining enzyme. If two pieces of DNA match against, ligase can link them to form a single unbroken molecule of DNA. Now, in DNA cloning, restriction enzymes and DNA ligase are used to insert genes and other pieces of DNA into plasmids. So, restriction enzymes are found in bacteria and other prokaryotes. They recognize and bind to specific sequences of DNA called restriction sites. Each restriction enzyme recognizes just one or few restriction sites. When it finds its target sequence, the restriction enzyme will make a double-stranded cut in the DNA molecule. Typically, the cut is near the restriction site and occurs in a tidy, predictable pattern. As an example of how a restriction enzyme recognizes and cuts a DNA sequence, let's consider EcoR1, a common restriction enzyme used in labs. EcoR1 cuts at the following site. DNA ligase. In DNA replication, ligase's job is to join together fragments of newly synthesized DNA to form a strand. So the ligases used in DNA cloning do basically the same thing. So if two pieces of DNA are matching, DNA ligase can join them together to make an unbroken molecule. Okay. Now if we talk about bacterial transformation, so plasmids and other DNA can be introduced into bacteria such as a harmless E. coli used in labs in a process called transformation. So during transformation, specially prepared bacterial cells are given a shock, like such as high temperature, that encourages them to take up foreign DNA. Bacteria can take up foreign DNA in a process called transformation. Transformation is a key step in DNA cloning. It occurs after restriction, digestion, and ligation and transfers newly made plasmids to bacteria. After transformation, bacteria are selected on antibiotic plates. Bacteria with a plasmid or antibiotic resistant, and each one will form a colony. So colonies with the right plasmid can be grown to make large cultures of identical bacteria which are used to make protein. The transformation of bacterial cells, a very inefficient process, a process for inserting foreign DNA into bacteria. Treat bacterial cells with calcium chloride, add plasmid DNA to cells, chilled on ice, heat the cell and DNA mixture. Plasmid DNA enters bacterial cells and is replicated and express their genes. Electroporation. Apply brief pulse of high voltage electricity to create tiny holes in the bacterial cell wall that allow the DNA to enter. Selection of recombinant bacteria transformations. So selection is a process designed to facilitate the identification of recombinant bacteria while preventing the growth of non-transformed bacteria and bacteria that contain plasmid with a foreign DNA. So plate transform cells on plates containing different antibiotics to identify recombinant bacteria and non-transformed bacteria. So we can talk about blue white selection in very brief. DNA is cloned into the restriction site of the LAG-Z gene. It's an operon, basically a part of the operon. So when it is interrupted by an inserted gene, the LAG-Z gene cannot produce functional beta-gal. So when egg-gal, that is artificial lactose, is added to the plate, if functional LAG-Z is present, blue colony, non-functional LAG-Z is white colony, because genetically identical bacterial cells each containing copies of recombinant plasmid. So these are the practical steps of bacterial transformation and selection. Typical procedure for doing that. Now, if we talk about protein production, how this links to our cytokine therapy. So once we have found a bacterial colony with the right plasmid, we can grow a large culture of plasmid-bearing bacteria. Then we give the bacteria a chemical signal that instructs them to make the target protein. The bacteria serve as miniature factories, basically churning out large amounts of protein. For instance, if a plasmid contained the human insulin gene, the bacteria would start transcribing the gene and translating the mRNA to produce many molecules of human insulin protein, right? So once the protein has been produced, the bacterial cells can be split open to release it. There are many other proteins and macromolecules floating around in bacteria besides the target protein, like insulin. Because of this, the target protein must be purified or separated from the other contents of the cells by biochemical techniques. Different types of chromatography. And the purified protein can be used for experiments, or in the case of insulin, can be administered to patients once the purity is ascertained, basically. Okay. So what are the uses of DNA cloning? DNA molecules built through cloning techniques. Built through cloning techniques are used for many purposes in molecular biology, like biopharmaceuticals. Right? DNA cloning can be used to make human proteins with biomedical applications, such as the insulin mentioned above. Or other examples of recombinant proteins include human growth hormone, which is given to the patients who are unable to synthesize the particular hormone, and tissue plasminogen activator, which is used to treat strokes and prevent blood clots. Recombinant proteins like these are often made in bacteria. Gene therapy: in some genetic disorders, patients lack the functional form of a particular gene. So, gene therapy attempts to provide a normal copy of the gene to the cells of a patient's body. For example, DNA cloning was used to build plasmids containing a normal version of the gene that's non-functional in cystic fibrosis. So, when the plasmids were delivered to the lungs of cystic fibrosis patients, lung function deteriorated less quickly. Then we have gene analysis. In basic research labs, biologists often use DNA cloning to build artificial recombinant versions of genes that help them understand how normal genes in an organism function. So these are just a few examples of how DNA cloning is used in biology today. DNA cloning is a very common technique that is used in a wide variety of molecular biology experiments. Okay, we will not go into too much detail. Earliest transgenic bacteria basically are used to produce human insulin for diabetes, like clotting factor for people with blood clotting disorders like hemophilia, and human growth hormone. So the human gene responsible for insulin production is cut from human DNA and inserted into a plasmid to make recombinant DNA. And the recombinant DNA is inserted into bacterial cells which are transformed and begin producing insulin. Now, transgenic mice have been produced with human genes that make their immune systems similar to humans, which allows scientists to study the effects of disease on human immune systems. The transgenic cows have been produced with extra copies of growth hormone genes that make them grow faster. Transgenic plants have been produced that contain genes which produce natural insecticides like insect killing chemicals or are resistant to weed killing chemicals but have a better flavor or texture. Okay. If we talk a little bit about from the plants section, it's a genetic potential of pluripotency. It's a genetic potential of a plant cell to produce the entire plant. So the basis of pluripotency is to stimulate. Basis of this is to stimulate pluripotency to grow large numbers of cells in a sterile controlled condition. Okay. We will not go into plant cell culture anyway. So the answer to your question is, how has genetic engineering impacted cytokine therapy? It has made it feasible to produce essential proteins for therapeutic purposes. Any questions? I'll wait for a few minutes. If you have any questions, you can ask me. Any questions? So also if you ask me in detail how genetic engineering has affected cytokine therapy with paper references and what people have done in research recently, then I can tell you that it has actually, genetic engineering has been such advantages for this, because if you want a research backed answer, uh, it has revolutionized cytokine therapy in three major phases. Number one is recombinant cytokine production. It's a foundational breakthrough. Earlier cytokines like interferons, IL-2 were scarce, very difficult to purify, heterogeneous. With recombinant DNA technology, cytokine genes can be cloned into expression systems like E. coli, yeast, even mammalian cells. And this enabled large-scale, pure and reproducible production. So the example can be recombinant interferons like interferon alpha, interferon beta, interferon gamma. They became widely available for antiviral therapy, cancer therapy, autoimmune disorders. Like recent work shows rational genetic design plus optimized expression systems can produce stable bioactive interferons with high yield and purity. So this directly validates why cytokine therapy became clinically viable. Number two is engineering cytokines to improve therapeutic performance. So a major limitation of native cytokines is that pleiotropy, like multiple effects, which can lead to side effects. Also short half-life and systemic toxicity. Modern genetic engineering strategies, recent reviews highlight these three key approaches. In order to reduce pleiotropy, they can modify the receptor binding regions. They can create biased cytokines like target specific pathways. Studies show engineering aims to deconvolute pleiotropism to improve specificity. The other approach is targeted delivery, like fusion protein, cytokine plus any kind of antibody. So it basically kind of doing tumor targeted interferons. So engineered interferons can be delivered specifically to tumor sites, improving efficacy and reducing toxicity. Then the other approach is improving pharmacokinetics like pegylation, Fc fusion proteins, nanoparticle delivery. So recent literature shows engineering improves half-life, stability and bioavailability. Now the other thing is the development of next generation cytokine therapeutics. So genetic engineering has enabled completely new classes of cytokine drugs like designer cytokines, modified IL-2 variants with reduced toxicity, selective immune activation. Like examples can be engineered IL-2 therapies aim to retain anti-tumor effects while reducing side effects. Interferon subtypes with improved safety like type 3 interferons, more cell-specific, fewer systemic side effects. So type 3 interferons show targeted anti-tumor activity with reduced toxicity. Then if we talk about cytokines in regenerative medicine, engineered cytokines can now be used in tissue repair and stem cell modulation. Cytokine engineering is expanding into regenerative applications. Then if we talk about integration with modern therapeutic platforms, like genetic engineering has allowed cytokines to be combined with immunotherapy, like CAR T cells engineered to secrete cytokines, tumor microenvironment modulation, gene therapy. Like cytokine genes can be delivered via viral vectors. It can be sustained. It can show sustained in vivo production. Then if we talk about the key scientific challenge, so all is not easy. There are tremendous challenges as well associated with these kind of work, because all of these will definitely go for clinical trials, right? So it's not that easy to qualify for clinical trials without the advantages outweighing the disadvantage or the limitations. So if you talk about the key scientific challenges that basically the current research is focusing on, basically cytokine toxicity, right? Cytokine storm risk, then off-target immune activation, optimal dosing and delivery, all of this. Then the recent studies emphasize need for precision cytokine engineering and balancing efficacy with safety. That is the need of the hour, but anyway, genetic engineering has anyway revolutionized this field for sure. Okay. So final summary, if you want, genetic engineering has transformed cytokine therapy by enabling large-scale recombinant production of cytokines. Number 2, allowing structural modification to improve specificity and reduce toxicity. Number 3, enhancing pharmacokinetics and targeted delivery. Number 4, creating next generation cytokine drugs, engineered interleukins and interferons. And number 5, integrating cytokines into modern immunotherapy and gene therapy platforms. Now, if you want some paper references that you want to go through, basically it's better that you go through some research papers where people are working on the on the bench, like they're working practically, not only reading, not only theoretical knowledge. If you want, I want you to give me, if you tell me that I want you to give me some paper references, then some paper, proper critical research, peer-reviewed research article references. This is exactly how you should think at this stage. So there's a 2024 paper, Engineered IL-2 Fusion Proteins, where researchers engineered the interleukin to fuse with an antibody, immunocytokine. So goal was to reduce toxicity and improve targeting, and they designed fusion proteins in IL-2 plus antibody and tested in preclinical tumor models and measured immune cell activation and tumor regression. And their key finding was preferential activation of effector immune cells over regulatory T cells, better anti-tumor activity with less toxicity. Then we have targeted cytokines to tumor vasculature. It was published in 2020 in OncoTargets. It's engineered a hybrid IL-2 IL-15 cytokine targeted specifically to tumor blood vessels. So they basically did protein engineering of cytokine targeting moiety added like antibody base and tested in tumor models. The concept is delivering cytokines only when needed. Tumor size reduces systemic side effects. Then we have cytokine delivery, right? Using engineered cells, like it's a Nature paper. They basically use genetically engineered mesenchymal stem cells, and these cells produce IL-2 inside the tumor environment. So what they did was they engineered stem cells, expressed IL-2 gene, implanted into tumor-bearing models. The result was reactivated CD8 plus T cells. It overcame immunotherapy resistance. Then we have reprogrammed IL-2 using synthetic biology. It's also a Nature Communication paper. IL-2 redesigned using non-natural amino acids like semi-synthetic organism. They expanded genetic code, created IL-2 variants with novel IL-2 variants with altered receptor binding. The importance was that precision control over cytokine signaling. Then we have interferon engineering for cancer therapy. It's in Biomedicine and Pharmacotherapy 2024. Basically, what they have done, this is engineered type 1 and type 3 interferons, modified interferon structure, tested tumor targeting and immune activation. The key insight was type 3 interferons are more selective, less toxic than classical interferons. Then we have cytokine engineering strategies, experimental translation. It is published in Trends in Biochemical Sciences, EMC review. It basically includes real experimental approaches, pegylation, fusion proteins, mRNA cytokine delivery, cell-based cytokine secretion, all of that. Hydrogel encapsulated IL-2 producing cells, increased local cytokine concentration, reduced systemic toxicity, all of that. And then we have systemic experimental landscape with multiple cytokines. It was published in Frontiers in Immunology. So basically they summarize experimental pipelines, IL-2 variants, IL-15 engineering, interferon modification. The key experimental findings were native cytokines have short half-life, high toxicity, but engineering solves targeting, stability and specificity. So basically that is what you can talk about. Read. These papers are practical implications of what people are doing in real life, not only based on theory but also based on practical examples. You can go through these papers as well for your knowledge. Any questions? Okay, so if you have no further questions, maybe we can stop the session for today over here and meet again next Monday, same time, same link. Okay. Okay. Thank you.